invite Dr. Mahesh Verma. Dr. Mahesh Verma is Associate Director of AHA Immune Biosciences, a clinical stage drug discovery company based out of Pune, Maharashtra. AHA Immune has developed a new chemical entity for the treatment of vitiligo, which has now completed phase 1 trials. At AHA Immune, Dr. Mahesh heads in vitro assay development, animal biology, and clinical development. He will be talking on small molecule AB1001 as a topical non-steroidal therapeutic approach for vitiligo. Dr. Mahesh, please.
Thank you. Thanks for the introduction. And very good afternoon to all of you. I'm actually very much excited to be here and present our work in front of you. And the success story of our company, where we have developed a small molecule as a new therapy, a new arsenal for you people to use for the vitiligo patients. So we are talking about the small molecule AB1001 is a topical non-steroidal therapeutic approach for the vitiligo. At this platform, I don't think I need to give any introduction about the vitiligo. But the disease pathology which is talked about, here I want to put a little bit emphasis on how the disease progression happens. It is believed that in the melanocytes, they get some sort of trigger, and the trigger leads to the cellular stress. Because of this cellular stress, melanocyte destruction happens. Melanocyte destruction happens. And during this destruction of melanocyte and elevation of the cellular stress, many of the internal protein of the cells, they come out on the plasma membrane, which basically act as an autoantigen, and the immunity against these protein is made. And the immune system comes, and this autoimmunity is developed. Now this autoimmunity based destruction of melanocyte happen, and we see a vitiligo patch getting formed, and bigger patches being formed. So right now the treatment which is talked about, like steroid or other immunosuppressant, and now the JAK inhibitor, which is the first FDA approved therapy for the vitiligo, they generally try to inhibit the immune cell activity without showing any effect on the stress which is present in the melanocyte. So our approach was to develop the molecule which can take care of this part. So we have made a series of small molecules. We have screened them for the stress response, and after a lot of in vitro and in vivo assays, we have found a lead molecule AB1001, which not only reduces the stress response in the melanocyte, which we have also shown that it reduces the melanocyte destruction. It also stops the melanocyte recognition by the immune cell, and we have shown that vitiligo patch in the animal model is reduced. So let's see its effect on the melanocytes. So we developed an in vitro assay where we provide the stress, and the melanocytes, they die. And in this background, AB1001 in a dose-dependent manner rescue the cells from the death. And it not only reduces the cell death of the melanocyte, it also increases their function, like the tyrosinase enzyme activity. I have put it here. This is the enzyme required for the melanin synthesis, which is increased by AB1001 in a dose-dependent manner. And this effect is not achieved by the steroid, corticosteroid, or by the ruxolitinib. Similarly, the gene expression for tyrosinase and TYRP1 is increased by the AB1001. And not only that melanin function, also the melanocyte marker by which the melanocyte identity is defined, they are also increased by the AB1001, like MTAF and DCT expression. After getting this data, we went ahead and did the animal study for the AB1001, and we know that there are not very good animal models are available. So we have developed a guinea pig model. The reason for choosing the guinea pig is that it has the melanocyte in the skin compared to any other animal model like mouse, where the melanocytes are in the hair. So these animals were induced with the vitiligo causing cream, which has the compound which destructs the melanocyte, and you can see that the depigmentation patch is increased. And this is associated with the decreased expression of the melanocyte marker, increase in the T cell marker, and the inflammatory cytokines. After that, we developed the formulation, topical formulation for AB1001, which we can apply on the skin or the ear skin of the animal. And also a control placebo formulation was developed. And this was applied on left and right ear on the same animal. And we can see that the AB1001 topical treatment lead to the better repigmentation. This visual quantification of AB1001 leading to the repigmentation, if we quantify by the image analysis software, we can see a very significant increase in the pigment level and a reduction in the depigmentation level. When we take out this ER and do a histopathology and try to measure the melanin level, we can see that the much more and enhanced amount of melanin is present, which is quantified and found to be very, very significant. And this AB1001 induced increase in the pigment is also associated with the increase in the melanocyte markers. All those gene expression is high by the AB1001. And also same time the T cell marker are also reduced and the inflammatory cytokines are also reduced, saying that the skin is having less inflamed nature. Then we compared whether this compound, our compound is how good is compared to the other marketed products. So we have taken a corticosteroid. Topically we applied on the left ear and AB1001 applied on the right ear of the animals. And in the same animals we have seen and we found that our compound, the AB1001, shows the better repigmentation. And melanin level, in the histopathological level, we can see a very significant increase compared to the steroid. So it tells that our compound is actually superior to the steroid, at least in the animal model. So the take-home data from the in vitro and in vivo models is that it can increase the AB1001 can increase the repigmentation. It can improve the histopathological score, upregulate the melanocyte marker, as well as it decreases the inflammatory cytokines present in the skin. It reduces the T cell infiltration and it's also superior to the marketed product. So after getting this molecule, which is very potent and efficacious, we developed it as a drug candidate. So for that, we first did the CMC, chemistry, manufacturing and control. We identified the synthesis process where the scale-up production in the multi-kg level can be achieved. We have also identified the analytical method. We developed that method and we control on the impurities. Similarly, we done the stability study and the long-term stability study and found that both the API as well as the topical formulation is very stable in normal and the accelerated condition. And to go ahead, we need to do a lot, show the toxicity profile. So we have done all the preclinical toxicity study in a GLP background. We found that AB1001 is not toxic. It has no dermal or oral toxicity. It's not genotoxic. It doesn't cause any mutagenesis or clastogenic. This is not phototoxic, and this compound is non-sensitizer. If you apply on the animal, there is no skin hypersensitivity at all. And it is very, very safe. If you give it to the very high doses to the animal for the 90-day period and see their systemic level and the skin level, there is no toxic effect was there. So based on these data, the in vitro data, in vivo data, the CMC, then the preclinical toxicity and pharmacokinetics data, we went ahead and filed for the IND application, investigative new drug application. And we get the approval for the phase 1 clinical trial in the healthy volunteer. And then we produced this AB1001 API and formulation in a GMP scale, GMP batches. Then we went ahead and did the phase 1 trial. The phase 1 trial was an open label trial with a dose escalation study, whereas multiple ascending order of the doses were given to the healthy human. The primary aim of this clinical trial was to evaluate safety and tolerability, and the secondary aim was to measure the pharmacokinetics in the adult, healthy adult volunteers. So in this phase 1 trial, in the PK data, among the many parameters what we study, one is that we study how much it is remaining in the system after repeated dose. And this data says that these levels are more or less achieved, so it does not accumulate more and more in the system. Means repeated dose can be given for the long term. And the analysis of the skin where the AB1001 is applied, it shows that there is no skin-related problem and there is no safety concerns are there. All hematological, biochemical tested are also said that there is no safety concern for this thing. So it is a very safe molecule to study with that. So in the summary, this is the journey of the immune. We started from scratch, from the small molecule synthesis in 2016, and in the 6th year, we got our molecule and which is very safe for the under the phase 1 trial. We are now ready for the phase 2 clinical trial, which we are going to do in vitiligo patient, and it's going to be a randomized controlled trial. So we hope after getting that efficacy, it's going to be a new therapy for the vitiligo patient. So let me thank here for my team and the CRO partners here. 4 of us are present here in this meeting. Thank you very much.
Thank you, Dr. Mahesh. And now the topic is open for discussion. We can take a few questions if they are there. Yes, please. Can we have the mic there?
Congratulations for this very, very long journey. Not easy to come up with a new molecule. Just looking to the future, as you develop your study design, have you thought about what your endpoint will be? Stabilization versus repigmentation? Have you thought about your type of inclusion criteria? What kind of patients will you recruit? But this is going to be key to deciding if your molecule works or not. So given your toxicity, what you know from the laboratory experiments, what are your thoughts towards that study design?
Very good question. Actually, we were thinking a lot about in this area. Thing is that what type of vitiligo patient to be included in that next trial and all. So right now, our aim is to take all type of the vitiligo without any segregations. The molecule which is working here, we believe that it will work better on the progressive vitiligo, especially on the newly formed patches. And we have many more data to believe that it will work on that. So if I have to evaluate these data together, it stops the depigmentation, and it also brings back the repigmentation. So it works in the both the things, which are both the side desired for the vitiligo therapy. Yes, sir.
Based on your animal model, how do you know that the medication reaches the lower levels of the epidermis where the oxidative free radicals are present, which your molecule is trying to reverse? And the second question is, what makes you sure that blocking the oxidation free radicals, the unfolded protein response, what makes you sure that all the millions of T cells that are actually causing the vitiligo will disperse and cause repigmentation? Were you able to see that in your animal model?
Yes, yes, sir. We can able to see that our data. This is the actually same questions we used to ask ourselves when we have started our journey. If we stop that stress in the melanocyte, whether the effect coming from the immune cell will it be stopped or not. But actually, we are seeing in that data that like you are seeing the T cell marker which is going down. So we are very, very hopeful for that, and hope we'll get the same data in that clinical trial.
Thank you very much. That was really, really great talk.
Thank you.
Congratulations. I was just wondering, in your animal model, did you look at any regulatory T cells? I mean, does your drug potentially have an effect on regulatory cells or memory T cells or anything like that?
Great, it's great. Actually, we have very, very preliminary data. I think it is immature of me to tell that we have done. There are some problems because of the guinea pigs are there. You don't get a very good antibodies and all those things. So there are limitations are there. But we are looking into it. Maybe in some sort of the time we will have very definitive answer to give for that.
Thank you.
If we don't have any other questions, we can then move on. There's one question at the back, please. I think this will be the last we can take for this session.
I'll be quick. Thank you for the presentation. So you said the molecule primarily acts on the stressor aspect. So I want to know what was the stressor component you studied in your preclinical models so we can extrapolate it to the human studies as well. And the second question is there a specific target that we are studying? Because this was kind of a broader term. Or the chemical nature, since you said it was a non-steroidal molecule. If you could elaborate on that, please.
Yes. To answer that, the chemical structure, it is it's a heterocyclic molecule. It's a very small molecule, less than 500 kilodalton. Its molecular weight. And to answer it, what sort of stressor we have seen it. So we don't study those more in that preclinical toxicity and all. We have seen those stressor, their level is more in the animal model and a lot in the cellular system. So we have studied the, like you said, the endoplasmic reticulum stress and oxidative stress and the stress causing from a high inflammation. So based on, because of the time we didn't put, but we have the data to show that in presence of very, very high inflammatory background, this molecule reverse the effect, deleterious effect which are there on the melanocyte.
I think we'll have to close this talk here because any new molecule is going to generate a lot of interest, especially in a disease like vitiligo. But due to paucity of time, we'll move on to the further talks.
Thank you.
Thank you, Dr. Mahesh. Thank you very much.